Our first speaker this afternoon is Dr. Larry Purcell. Dr. Purcell comes all the way from the University of Arkansas, where he's a distinguished professor of crop physiology. Larry focuses on optimizing crop use of light, water, and nutrients through management and genetic strategies. Dr. Purcell received his PhD from the University of Florida. Larry.
Thanks so much, Jim. I really appreciate the opportunity to be with you here this afternoon and the invitation from Jason and Jim and the executive committee on the invitation to join this meeting. So it's been interesting as I've started looking at tropical agricultural production. I work almost exclusively in soybean and a little bit of corn work. And when I started preparing this talk and looked at the diversity of different crops that... Okay, clicker, here we go.
Is it working?
Somewhat. The technical difficulty band, I think, is joining us again.
Oh, I'm in the way. My fault.
God.
Is the clicker on? Do you see a signal on the top of it in the window?
It's working a little bit. Okay. So when I started looking at the diversity of tropical crops and the idea that I was going to be speaking about how climate change may affect tropical crop production, it was a little bit overwhelming. And boy, this is just going to be very difficult.
Why don't you just tell her and she can do it? Sorry, Larry.
Okay. We need to work out some signals here, some signal form or something. Can you back up one? Okay, so it really is three talks. And it's kind of a combination of climate change, genetic diversity, and tropical crops. And more specifically, the outline for my talk is given here. What can we expect from climate change? What impact will climate change have on crops? How can genetic diversity improve crop resiliency? And then we'll look at a couple of historical examples, and then we'll look at some of the work that I've done in soybean related to drought response. And then we'll look at three examples of tropical crops and how all of these three different things of climate change, genetic diversity may affect them. So with that in mind, what can we expect from climate change? This is some work that was done by Wang et al., recently published. We're looking here at projections from 2065 to 2099 versus kind of the baseline from 1979 to 2013. And I've got outlined in red kind of the tropical region from Tropic of Cancer, Tropic of Capricorn. And that region in there that you see with blue or green is projected to have an increase in the average rainfall per day. Now, what that means is that just the frequency and the intensity of extreme monsoonal rainfall is projected to increase. And somewhat counterintuitive, this will likely result in an increased risk of drought in some regions, and that's because the rainfall is not going to be evenly distributed around for every day of the year. Monsoonal rainfall is projected to increase in South and East Asia and equatorial Africa and decrease in North America and be unchanged in most of the southern hemisphere. We can do the same thing if we look at surface air temperature. And in this case, the tropical region there, you can see, is pretty much thoroughly outlined in red. These projections estimate that we'll be looking at about a 3.5 degrees centigrade increase over land, and that the intensity and frequency of the hottest days will increase. There it is. And for the 5% of the hottest days, they're projected to be 20% hotter than the mean projected temperature increase. So the hottest days are going to get even hotter than that projected 3.5 degree temperature increase. So how will climate change affect tropical crops? Let's just go through some of the different scenarios based on this. The increase in rainfall intensity, the increase in temperature, and the increase in dry periods because of this rainfall being unequally distributed will almost certainly increase in drought stress in certain regions. The increase in rainfall and humidity plus higher temperatures makes the perfect storm for increased disease pressure. The increase in temperature will also have an effect on different insect pests. There's likely to be an increase in the geographic range of certain insects, the increase in the number of generations per year, an increase in insect-transmitted disease, and it's unclear, but there's some projections that biological control will be impacted because beneficial insects may be impacted more than pest insects, and predation may also be affected. How will climate change affect crops? We've got the drought effects, and we also have flooding effects. Increase in rainfall intensity may increase the flooding stress, hypoxia, and everything that goes along with it. The increase in runoff may be impacted, resulting in nutrient loss and erosion. And because water's not infiltrating very well into the soil, when you have intense rainfall events, it may decrease plant available water, that wetting the soil profile. So I want to turn now to a couple of historical examples, and then we'll look a little bit at some work that we've done in soybean, looking at genetic diversity and response to drought. So from a historical example, let's look first at the Irish potato famine. This is certainly something that I'm sure everyone is at least tangentially familiar with. From 1847 to 1852, 1 in 8 people in Ireland died from starvation, and about that many people left Ireland seeking better situation. The causative pathogen of this was a Phytophthora species. The predominant variety were all clones from the variety Lumper. Soon after the foliage was affected, the tubers, within days, the tubers were rotting in the field and inedible. So a very, very rapid response. And again, we're looking at a variety that was essentially a clone throughout Ireland. More recently, the southern corn leaf blight, 1970, 1971. This was a familiar sight during the 1960s, early 1960s, when people would go out and detassel the corn. If you think about by 1960, about 90% of the corn were U.S. hybrids, and so you had to detassel the corn on the female parent in order to produce the hybrid seed, or otherwise you'd get self-pollination. So scores of people from Iowa and Illinois in high school, their summer jobs were clipping the tops out of corn. So yeah, this hybrid seed corn production required the manual removal of the tassels. And in the mid-20th century, it was discovered that there was something called cytoplasmic male sterility. And in this case, the female parent, the pollen was sterile. And so if it did fall on the silks, you did not get pollination. So you'd have the male parent planted next to the female parent with cytoplasmic male sterility, and you did not need to detassel the corn. So this was a huge boon. It decreased the cost of producing hybrid corn. There was a problem with this, though. By 1970, about 75 to 90% of the hybrid corn used this cytoplasmic male sterility, but the cytoplasmic male sterility made the corn susceptible to southern corn leaf blight. So on your left there, you see the hybrid that has this cytoplasmic male sterility, and on the right is hybrid that resulted from manual detasseling. So there's a huge effect on the corn. The blight destroyed about 15% of the U.S. corn crop, but local losses sometimes exceeded 30 to 80% of the corn that was destroyed by this disease. What was the common feature between the Irish potato blight, and the southern corn leaf blight? Of course, that's a lack of genetic diversity. Soon after this, in 1972, a really interesting review article was published in the Annual Journal of Plant Phytopathology, and I'll read a couple of sentences from that. Never again should such a major cultivated species be molded into such uniformity that it is so universally vulnerable to attack by a pathogen, an insect, or environmental stress. Diversity must be maintained in both the genetic and cytoplasmic constitution of all important crop species. So with that, I want to turn our attention now to soybean. And soybean, I know a little bit more about. Soybean is, the USDA germplasm collection has around 19,000 accessions of glycine max and around 1,200 accessions of glycine soja, the wild progenitor species of soybean. Despite this, soybean in U.S. varieties has a very narrow genetic base. There are around 35 genotypes that contribute around 95% of the genes, but these are not equally, each genotype does not equally contribute the same number of genes. Around 10 genotypes contribute 80% of the genes, and from the southern germplasm, 2 genotypes contribute 50% of the genes. So we have a very narrow genetic base of varieties. Most of the U.S. varieties result from elite by elite crosses. So you have the same genetic background basically, and you're just reshuffling those same genes. And for my research, my particular interest is there's very little difference in drought tolerance among these varieties. And so the USDA and the United Soybean Board recognized that this lack of genetic diversity limited progress and increased the vulnerability of the soybean crop as a whole to both biotic stress, abiotic stress, and yield advancement. So they made a major investment in genotyping all 20,000 genotypes in the collection with 50,000 molecular markers per genotype, SNP markers. So my interest was in identifying physiological traits that confer drought tolerance. And we focused on 4 traits: delayed canopy wilting, carbon isotope ratio, which is a surrogate measure of water use efficiency, aerial canopy temperature, and nitrogen fixation. So let's look a little bit at those 4 traits. Why these traits? For canopy wilting, this has been successful in other crops. If you're familiar with Aquamax corn, it was originally discovered that the differences in this and the original Aquamax germplasm was due to this slow canopy wilting trait that was visually observed. And it's a relatively quick, low-tech way of phenotyping plants. C13 ratio, as I mentioned, this is an integrated measure of water use efficiency. It also has a very high heritability, which makes it attractive from a breeding perspective. Canopy temperature, under drought stress, cool plants indicate that the plants are still transpiring water and they have access to soil water, soil moisture. And you can think about this as similar to canopy wilting in some cases, but it's less subjective than wilting. And then nitrogen fixation. Nitrogen fixation is particularly sensitive to drought stress, and it's closely related to yield. So these were the 4 traits that we focused our attention on. We used genome-wide association studies in evaluating this, and this is not going to be a seminar on genetics or on genome-wide association studies, but just to give you an idea of what we did, we had a panel of 373 very diverse maturity group 4 sessions and a handful of checks. We evaluated these in 4 different environments, and for each accession, we associated each of those 4 traits with each SNP that had been marked on in the germplasm. So this is an example of canopy wilting where we're looking at a frequency distribution. So we've got the frequency on the y-axis and an average canopy wilting score on the x-axis. So 0 would be no canopy wilting, 100 would be basically the plant's dead, it's wilted so badly. So we have a wide distribution of canopy wilting scores. We have 2 genotypes here. The one right there in the middle, PI 416937, before this work was done, it was considered the slowest soybean accession for canopy wilting. And then the ASGRO 5959 was the fastest genotype that we had found for canopy wilting. So we've got things that are more extreme than either of our checks from this panel. It had a high heritability, heritability of 0.80, which is really quite good. And this is kind of a molecular map of what we found. On the y-axis are the 20 different chromosomes in soybean, and on the x-axis is the base pair position on the chromosome. And the little black dots, they're a little bit difficult to see, but if it looks like a line, that's because those represent molecular markers that are very, very close together. And so each black dot represents a different SNP or molecular marker. The red bars above the chromosomes represent places in the germplasm, in the genomic positions where we had a slow wilting association that was significant. So each red bar represents, you can think of this as a gene or an allele that's associated with slow wilting. There were 61 SNPs, and these were categorized into 46 different loci. The next trait is C13 ratio. Again, this is a surrogate measure of water use efficiency. The frequency distribution here is shown very similar. We've got a very wide distribution. We had a broad sense heritability of 0.76, again, quite high. We found 54 SNPs significantly associated with C13 ratio, and this was in 46 different loci. The next trait that we looked at was canopy temperature. We measured canopy temperature using a drone with an infrared camera associated with it, a very lightweight camera that had its own digital recorder attached to it. We would fly this above our soybean field, and from that we were able to distinguish the canopy temperature. The infrared camera, in an image I'll show you, white is hot and black is cold. It's able to detect temperature differences of 0.05 degrees centigrade. This is a picture of my cat taken with the infrared camera. What I want you to notice is if you look very closely at the bottom of the screen, you'll see 4 little white dots. Those represent the cat footprints. As it walked across the floor, it exchanged enough heat to show up on the floor, very distinguished just from a a second as it walked across the floor. So that gives you an idea of how sensitive this camera is. This is a picture of a soybean field. Each one of those black rectangles or gray rectangles represents a different soybean genotype. And so there's 256 different shades of gray from white to black in here that corresponds to a temperature. The white that you see between each plot is the soil temperature that's radiating heat. This is a frequency distribution of our canopy temperature. Again, we have, it's normalized, so it's not in actual degrees centigrade, but what you can take from this is there's a broad range of differences among genotypes for this average normalized canopy temperature. The heritability on this was somewhat lower, 0.5. We found 52 SNPs that were significantly associated with canopy temperature, 34 loci. So again, this is the kind of map that we use to identify these significant SNPs on each of the chromosomes. So the blue and green bars that you see there represent these significant molecular markers associated with canopy temperature. And 15 of these positions that we found for canopy temperature were common with wilting, the slow wilting, canopy wilting trait. So that really actually makes a lot of sense, that if you have something that's slow wilting, it may also be able to transpire when drought does occur, resulting in cooler temperatures. Okay, we're going to look, this is the last trait that we'll talk about. This is looking at nitrogen fixation. Soybean is, of course, a legume and fixes its own nitrogen from the atmosphere and the root nodules that are infected with Bradyrhizobium japonicum. The genotype on the right is a mutant that is unable to fix nitrogen, and so it's very nitrogen deficient. It gives you an idea of how much nitrogen is being derived from nitrogen fixation by the genotype on the left. We have a wide range of nitrogen fixation among these genotypes. The heritability is quite a bit lower, 0.35, and it had 12 putative loci associated with it. So let's look at the favorable markers that we have from this GWAS study. Canopy wilting, we had 61 SNPs, molecular markers, C13 ratio 54, canopy temperature 52, and NDFA or nitrogen fixation 64, all total 231. If there's breeders here, I imagine they're shaking their head like this is a genetic nightmare. And so to recover all of these favorable genes or alleles is almost impossible. And so you would have to have, I don't even know, trillions and billions more in a field of individual plants to recover the one or two plants that has all of these favorable alleles for these four traits. But we can take some shortcuts. And this is looking at accessions in the USDA germplasm collection with 60% or more of these favorable markers. So we've taken a subset of the USDA germplasm collection and we've identified those favorable markers, and then we've queried the entire 20,000 accessions in the germplasm collection to find those ones that have the most favorable alleles. So in the case of canopy wilting, there were 713 accessions that had greater than 60% of the favorable markers. For C13 ratio, there were 943. For canopy temperature, there were around 2,100. And for NDFA, there were close to 12,000. And if we look at how many accessions had 60% of all four of these favorable markers, there were 27. So what we did is we went back to the germplasm collection, and for each of these four traits, we queried the germplasm collection to identify the accessions that had the most, whether it was 60% or 80%, whichever accession had the most of these favorable traits for canopy wilting, for canopy temperature, for C13 ratio, or NDF, or for nitrogen fixation. We pulled those out of the germplasm collection, and we created a mating scheme or crossing scheme. So first we crossed an elite line, shown here with the EL, the green, times a genotype that had favorable alleles for canopy wilting. We also crossed this elite line for a genotype that had most of the favorable alleles for canopy temperature. Did the same thing for an elite line by a genotype with C13 ratio. I have it as WE for water use efficiency. And for an elite line times a genotype that had favorable alleles for nitrogen fixation. We then went and crossed the F1 plants, the progeny of that initial cross, together, so that in what's labeled there as G2, we have canopy wilting and canopy temperature alleles combined, and we have water use efficiency, C13 ratio, and nitrogen fixation alleles combined. And then in the next generation, we made that cross again with the F1 by F1 in the G2 generation to put all those in one genetic background. And so we had lots, in the G3 generation, we had lots and lots of these different crosses, individual crosses. We've selfed those, and we're now doing what they call genomic selection and phenotyping. Genomic selection means that we're going back to individual plants, and we're identifying how many of those favorable alleles for each of those 4 traits are in each plant. And we're using that to advance the next generation. So I know that we're talking about something that's very different when we're talking about many tropical plants. In my work in soybean, we've got it easy. We have a crop that has great genetic diversity. We have great molecular information about each of the accessions. It's an annual plant, so we can rapidly progress, make rapid progression, and it's self-pollinated. All of those make the job that we're trying to do much, much easier for something that is a perennial, that is slow generation times, and very often is something that outcrosses very much. So we've got it easy, but I hope that some of the things that I'm talking about will be transferable to some of the tropical crops that are important here for the tropics. So let's look at coffee. Arabica coffee has a very narrow genetic base. It's a tetraploid from a single hybridization event between 2 different coffee species. Geneticists will talk about this being a genetic bottleneck, that it makes it very difficult to go back and recreate that original Arabica type coffee that was generated. It has a relatively narrow optimum temperature range of around 18 to 21 degrees centigrade. The increased temperatures that we're thinking about, that I showed you earlier, projected for the tropics, is going to make coffee production move to higher and higher elevations. It's estimated that by 2050, we will lose about 50% of the current regions that are now acceptable for coffee production. Higher temperatures will exacerbate coffee rust and coffee berry borer, and maybe some other disease and insect pressures. These increased temperatures and drought may lead to decreased yield and quality. There are lots of different coffee collections around the world, germplasm collections, but my understanding is that some of these collections are restricted from free exchange. We heard some information this morning that this genetic coffee collection is expanding. I think that's a very positive thing. Preservation of the wild coffee species is necessary. It's estimated that about 60% of the coffee species, the genus, face extinction, and about 45% of these species are not, of these accessions are not in collections. Okay, there's about 19 accessions in the coffee genus, and 10 Arabica species in Hawaii have been identified as nematode resistant, and molecular marker data are being developed for coffee. Macadamia nut, it's a very recent domesticated crop. The first orchards were established in Hawaii in the 1920s. From these five cultivars were selected from around 20,000 different open pollinated seedlings. And most Hawaiian cultivars are just 2 generations away from the wild macadamia nut. Similar to coffee, the daytime temperatures for macadamia are around 10 to 26 with a pretty narrow rainfall of 120 to 300 centimeters per year. Increased temperatures and decreased rainfall may be detrimental to macadamia yields. In macadamia, we've talked a little bit this morning. We heard a little bit that grafting was very important for the production of this crop. This results in very little genetic diversity. Wild macadamia in Australia is being threatened by forest clearing. There have been some work on genotyping 3 different species with fairly high density molecular markers. 112 different genotypes have been fully sequenced. So a lot of this information is coming together. There's relatively little information on differences in the response to temperature or disease or other stresses, however, and that's kind of the other side of the puzzle. You have to not only have molecular marker information, you have to also have physiological information on phenotypes. Banana is something that I think everyone is very well aware of. And the mainland U.S., the only thing that you can find pretty much is Cavendish. Prior to 1950s, as many of you are certainly aware, Gross Michael was the prime variety, but it was susceptible to Panama disease. Cavendish is highly susceptible to a fungal disease, Sigatoka, and also to a new race of Panama disease, tropical race 4. And because banana is seedless, Cavendish is propagated vegetatively, so everything that we have out there is a clone. Banana is a very diverse genus. There are lots of different collections. A collection in Belgium has around 1,500 different varieties. There's, the genus has also been molecularly mapped with around 35,000 different molecular markers. There have been markers identified for resistance to tropical race 4, and there's been wild banana that's identified to resistant to Sigatoka, the wild banana's Calcutta 4. And there's been a gene identified from tomato that may offer resistance to Sigatoka if it is transferred to banana. One of the real success stories is certainly with papaya. Papaya production in Hawaii was devastated by papaya ring spot virus, and foundational work here at the University of Hawaii and other locations transferred resistance to papaya. And we have now rainbow and sun-up cultivars that have really saved papaya production in Hawaii. One concern that came up is that we need to expand, or there's a need to expand papaya ring spot virus resistance to other papaya, not just rainbow and sun-up, but have diverse sources of diverse germplasm that's resistant to papaya ring spot virus, but also just to have diverse cultivars that are developed that may be able to address other stresses that come about unexpectedly. So my take-home messages today that I'd like to share with you are climate change is coming. It's here. Everyone's aware of it, I think. It's going to bring increased temperatures, rainfall intensity, drought, disease, and insect pressure. Crop vulnerability to climate change increases as genetic uniformity increases. And germplasm needs to be preserved and characterized to target environments. And breeding for diversity can be augmented by molecular tools, and there's a need to really characterize our germplasm for specific traits that we believe are going to be important in the future. Thank you very much. It's been a pleasure to be here with you. I'm looking forward to our presentations this afternoon and our discussions and the panel discussion.
I have one question from the audience for Larry, and I think this is a really interesting one. Dr. Purcell, you make the point that a narrow base of genetic diversity can be highly problematic. What are the reasons that we see such narrow genetic bases in crops?
There's a couple of reasons, and there are probably geneticists here in the audience that can answer this better than me. But one reason that you have a narrow genetic base is that when people find something that yields well and works, they stay with it. If you have two genotypes that are high yielding, have good disease packages, and you want to create something else, you cross those two genotypes together. But when you do that over and over again, you narrow the genetic base. And that's one of the primary reasons. There are other reasons, but I think that that is one of the prime considerations.